Good afternoon. My name is Tiago Pereira. I'm from Portugal, University of Coimbra. My research work focuses on exploring deep reinforcement learning for the computational generation of molecules with optimized properties. In this specific case, the objective is to find molecules that can interact with the central nervous system receptor, which means that they need to permeate across the blood-brain barrier. The process of placing new drugs into the market comprises several stages. On average, the drug development process can take 15 years to complete, and about $1 billion can be spent over that period. In addition to time and high investment, another problem is the high risk of rejection by the competent authorities if the candidate drug does not comply with all the requirements. Different computational approaches have been implemented to streamline the drug development process by optimizing specific molecular properties. However, these approaches are often insufficient since drug candidates must contain a set of biological, physical, and chemical properties simultaneously optimized. In this work, we consider two essential conditions for the effective action of a drug. On the one hand, we aim to optimize the biological affinity towards the target, and on the other, its ability to reach the receptor, which means optimizing properties such as permeability and solubility. It should be mentioned that there are different formats to represent molecules. In this work, we use the canonical smiles, which are line notations to describe the chemical structure of the compounds. Our principal purpose was to generate a set of promising lead molecules in the form of smiles notation that should be capable of binding to the target and penetrate the blood-brain barrier. The intermediate steps to reach these goals are training a generator of valid molecules and models to predict the molecular properties to be optimized. Afterward, we implemented the reinforcement learning dynamics by integrating the previously trained models and applying a multi-objective optimization strategy to consider both objectives. Finally, we performed the evaluation of the obtained compounds. So this is the general overview of this work. The dynamics of generation and optimization start with the sampling of new smiles by the generator models. These molecules will be evaluated by the predictors, and based on the prediction of both properties, each molecule will have a reward. Finally, the combined reward will have two roles: adjusting the exploration-exploitation strategy and retraining the generator using the policy gradient algorithm to explore the regions of the chemical space that maximize the reward. The generator is a deep learning model composed of an input layer and two long short-term memory layers that process the one-hot encoding vectors. Also, its architecture contains a dense layer with a softmax activation to transform the output into a probability distribution, indicating the probability associated with each character to be selected. The molecules are constituted by tokens, and throughout the training process, they are analyzed token by token. Hence, the next character is predicted taking into account the previous structure, and the objective is to maximize the probability of choosing the correct character at each step. To do this, it is necessary to minimize the cross-entropy loss function, that is essentially the negative log likelihood ratio between the correct and the other tokens, to ensure that after each training step, the correct output token is more likely to be chosen in future generation steps. Regarding the biological affinity property, the target of interest was the adenosine A2A. The predictor was trained with a dataset containing approximately 5,000 molecules composed of the SMILES representation and the respective PIC50. This PIC50 measure stands for the negative logarithm of the half maximal inhibitory concentration. Hence, the objective is to maximize this parameter in the generated molecules so that they have greater potential to be antagonistic inhibitors of the receptor. Moreover, the regression was implemented with a recurrent neural network architecture, which intended to associate each molecule with its affinity for the target. In the case of the blood-brain barrier permeation predictor, it was not possible to directly find a robust dataset to train a deep learning model, since the available datasets contained few instances and were strongly imbalanced. The solution was the combination of different datasets and the application of oversampling techniques to balance the classes. In this case, the predictor was implemented with a dataset of more than 4,000 molecules containing the SMILES representation and the indication of the blood-brain barrier permeation or non-permeation. Therefore, we perform a study about the different aspects of the model, namely in terms of the oversampling technique, smooth or adenosine, the descriptor type, the extended connected fingerprint or SMILES, and the proposed architecture, recurrent neural networks or deep fully connected neural networks. In this case, we sought to implement a deep learning-based classification model to learn the structural characteristics of molecules that influence their permeability. Then, we implemented the reinforcement learning setting to generate molecules. Typically, the reinforcement learning dynamic is composed of an agent that, at each step, is in a given state of the environment in which it has to choose an action. As a result, the environment will change depending on the action, and the agent will be presented with a new state in the next step. Moreover, the agent will also receive a reward that will be as higher as better was the selected action. This dynamic was transposed to the generation of molecules. In this case, we have the generator sampling new molecules, while the predictor is working as an environment that evaluates the generated molecules. Depending on the compound's optimization, the generator receives more or less reward, and its weights are adjusted through the policy gradient algorithm accordingly. Thus, the objective function is defined to retrain the initial generator in order to maximize the reward obtained at each step. This function analyzes the probability of the generator choosing the next token Sj considering the remaining generated structure. The reward value indicates to the generator the actions that must be taken considering the previous context. In practice, the gradient of this function is computed and the generator weights are updated. The reward guarantees that the weights are biased more strongly in the direction that provides the generation of molecules with the desired molecular properties. In this sense, it is possible to guide the generator to different areas of the chemical space until finding those that provide greater rewards. As previously mentioned, we have considered two objectives. Therefore, the predictors will output a vector with two values as rewards that must be converted into scalar to be employed in the policy gradient algorithm. We examined two scalarization methods, the linear weighted sum and the Chebyshev. In both cases, it was necessary to previously define a weight that indicates the importance attributed to each one of them. In the linear weighted sum case, the objective is to perform a weighted average of the two rewards. In the Chebyshev scalarization, it is necessary to define an optimal point for each objective, and at each step, the method will employ the reward from the objective that is farthest away from the previously defined optimal reward. The initial generator was trained with about 500,000 molecules to learn how to generate valid molecules using the SMILES notation. Therefore, the model evaluation metrics were the rate of syntactically valid and unique molecules, as well as the Tanimoto diversity. This parameter varies between 0 and 1, where 0 indicates that the compared molecules are the same. It analyzes the extended connectivity fingerprint vectors of molecules to estimate the diversity between compounds of the same generator, the internal diversity, or to compare different sets of molecules with the external diversity. The model is capable of generating new valid molecules and not just mimicking the training dataset, as it is demonstrated by the 93% of valid molecules and the high Tanimoto diversities, as well as the percentage of unique molecules. The biological affinity predictor was implemented as a regression task. Therefore, the parameters employed to evaluate the model were the mean square error, the coefficient of determination, and the concordance correlation coefficient. the results demonstrate a decent predictive ability throughout the entire range of binding affinity values. For the permeability predictor, after collecting about 4,500 molecules, it was necessary to apply oversampling techniques, as only 750 molecules belonged to the class of non-permeable molecules. The selected techniques to generate new instances of data were the SMOTE and the ADASIN. Basically, the methods identify the points that belong to the minority class, and in the case of the SMOTE, new instances of data are generated in the lines that connect these points. For the ADASIN, the principle is the same, but it applies a mechanism to ensure a greater generation of new points in the regions with higher majority class density. For the blood-brain barrier predictor, we evaluated the most commonly used metrics for classification tasks. The results demonstrate that the molecules that can cross the blood-brain barrier are more easily identifiable since sensitivity is higher than specificity for all experiments. This is an expected result, as the initial dataset has far fewer examples of the blood-brain barrier non-permeable molecules. Oversampling techniques aim to minimize this tendency through synthetic data generation, but do not eliminate it entirely. Nevertheless, the highlighted model, implemented with the extended connectivity fingerprints and a fully connected neural network architecture, outperformed the other approaches. The purpose of the multi-objective optimization step was to determine the combination of weights that guaranteed the best compromise between the compounds' biological affinity and their permeability across the membrane. For both scalarization techniques, the weighting strategy was to perform a uniform sampling between 0 and 1 with a 0.1 step. These figures represent the solutions obtained in terms of rewards for each property at the end of the reinforcement learning process. In blue are represented the dominated, and in red the non-dominated solutions. The non-dominated solutions are of particular interest as these policies have optimized both objectives better than the dominated ones. In this regard, the non-dominated solutions were directly evaluated based on the characteristics of the generated molecules. Thus, the policy that guaranteed the best trade-off between all the desired properties was obtained when applying the Chebyshev scalarization with 0.5 for the weights of each one of the objectives. This result is supported by this figure, which depicts the difference in the PIC50 distributions of the molecules generated by the unbiased generator in red and by the optimized generator in green. As the molecules exhibit a higher PIC50, they will have greater potential to inhibit the receptor. Furthermore, the optimized model was able to generate a high percentage of molecules as likely to cross the blood-brain barrier according to the permeability predictor. We also analyzed other essential properties for lead molecules, such as their synthesizability and drug likeness, depicted here in figure 6a. Furthermore, we looked at properties that influence its ability to permeate the blood-brain membrane, such as the molecular weight or lipophilicity, depicted in figure 6b. In both cases, most molecules fall within the regions considered as desirable for these properties, which validates the applied approach. In summary, it was possible to develop a de novo molecular generation framework to make the process of identifying lead molecules more effective, considering the ambitious goal of creating molecules from scratch likely to cross the blood-brain barrier. The limitation of this work is the fact that it has guided the optimization process based on a model trained with a possibly scarce and imbalanced dataset, despite the strategies applied to make it more robust. Nevertheless, as depicted here, it was possible to find structural similarities between the molecules obtained from the model and some compounds known to inhibit the adenosine A2A receptor. In these three cases, as the structural similarity is evident, the functional similarity can also be deduced. Hence, we were able to generate molecules with two essential optimized objectives: the biological on-target affinity and the permeability across the blood-brain barrier. As a result, it was computationally possible to get closer to the real complexity of this problem. So, thank you for your attention, and now I'm available to answer all your questions about this work. Thank you.